Question 1 in Chapter 1. The common ancestor of all lives likely belongs to which domain? You know that the common ancestor for everything that lives on the earth is bacteria. And we know that bacteria has its own domain, that is domain bacteria. Second question, what is an emergent property? An emergent property is a property that's found in this individual, but not found in the components that's making the individual. Number 3, you should be able to identify independent, dependent, and controlled variables if I describe one experiment. So basically, the independent variable is the variable that you want to test, that you manipulate. You will change the conditions. The dependent variable is the parameter that you can measure, you can quantify. Controlled variables, they're also called standardized variables. These are the variables that can possibly make a difference to the experimental result. So therefore, you want to make sure that everything that you test will receive the same condition. For example, if you are testing which fertilizer is most effective for your tomato plants, the different brands of fertilizer will be the independent variable. The dependent variable will be the yield of your tomato plants, and the control variable could be the temperature, the amount of water that you give to the plants, the location where you grow them, etc. Chapter 2, question number 1. What is an atom made of? An atom is made of protons, neutrons, and electrons. What components are responsible for the atomic mass? The atomic mass is the sum of the protons and neutrons. This is because electrons almost weigh nothing. Question 2. What type of chemical bonds is due to atoms giving away electrons? The answer will be ionic bonds. The two atoms that form ionic bonds, they do not share electrons. One will give away, the other will take completely. What if they share electrons? That would make the chemical bond a covalent bond. And remember that if the two atoms share electrons equally, that would be called a nonpolar covalent bond. If they share not equally, that would be a polar covalent bond. Question number 3, how does a hydrophobic molecule behave in water? So here the main point is to be able to tell the difference between hydrophobic and hydrophilic. Hydrophobic means that the molecule does not like water, so therefore all these molecules will stay together, will hide away from the water. They form these little droplets. Number 4, what pH range would an acidic solution have? Acidic solution must have a pH higher than 0 but smaller than 7, so from 0 to 7. What ions is it rich in? Acidic solutions, they have a lot of protons or H+ or hydrogen ions. Number 5, what's the scientific name for the human species? That would be Homo sapiens. There's a S at the end of the species name. Remember that when you write scientific names, you must italicize every letter if typewritten, but if you handwrite the names, each word must be individually underlined. And also, the only letter in the name that can be capitalized is the first letter of the genus name. So, Homo sapiens, only the letter H is capitalized. Number 6, how many electrons can each energy shell hold? You are only required to remember the first 3 energy shells. The first one can hold 2, second one can hold 8, the third one can hold 8 as well. What is the valence shell? The valence shell is the outermost shell of any given atom. How is the stability of an atom determined by its valence shell electrons? So basically, if the valence shell is completely filled or completely empty, then the atom will be more stable. So therefore, for example, if you think about magnesium, which have which has 12 protons. So normally it has 12 electrons as well. So you put 2 in the first energy shell, 8 in the second energy shell, 2 in the valence shell. However, that's not stable because it's not completely filled. So therefore, magnesium actually tends to lose those 2 electrons. Chapter 3. What are the monomers for proteins, carbohydrates, and nucleic acids called? Monomers for proteins, they are the amino acids. For carbohydrates, that's the monosaccharides. For nucleic acids, that would be nucleotides. What's the biological molecule that make up the cell membrane? That would be phospholipids. Remember that the cell membrane is a phospholipid bilayer. That would be the major component. Number 3, what are main storage carbohydrate chemicals in plants and animals? Remember that in plants use starch to store the carbohydrates, or in animals that would be glycogen. Chapter 4. Which eukaryotic organelle specializes in energy production? That would be the mitochondria. Number 2. What cellular structures are found in all organisms? That means bacteria, plants, animals, fungi, everything. The common structures include cell membrane, ribosomes, DNA, and the cytoplasm. All the other structures can be found in some organisms, but not the others. Number 3, what organelles have their own DNAs? So first, nucleus has DNAs, and then remember that mitochondria and chloroplast, they used to be this free-living bacteria, at least according to this endosymbiosis theory, and they both have their own DNAs as well. So 3 organelles, the nucleus, mitochondria, and chloroplast. Chapter 5. How is potential energy different from kinetic energy? And give a few examples. Potential energy is the energy that is stored, that is not being used. Kinetic energy is the energy in use. Think about motion, right? So therefore, one example of a potential energy, that would be the chemical bonds in a chemical, the bonds that's stored in food, for example. Think about a water dam. Think about the bicycle on top of a hill, ready to go down. Right? Those are potential energies. Kinetic energies include light, sound, radiation, you pedaling your bicycle, a fish swimming, etc. Second question, what is negative feedback? So negative feedback refers to these metabolic pathways where the product from the lost enzyme will go back and inhibit the activity of the first enzyme, right? That's the negative feedback. The product going back to slow down the activity of the first enzyme. What's the difference between competitive and non-competitive inhibitors? So basically, competitive inhibitors, they will bond to the active site, compete with the actual substrate, therefore making the enzyme less efficient. A non-competitive inhibitor, on the other hand, can make the enzyme completely useless because a non-competitive inhibitor will bond to a different location other than this active site. And when that happens, that will change the shape of the active site. Therefore, the substrate cannot bond at all. Number 3, what is the definition of metabolism? Metabolism is all the chemical reactions that happens inside a cell. So it includes those reactions that can build up larger molecules, and it also includes reactions that will break down those complex molecules to smaller and simpler molecules. Some reactions release energy, others create energy, but everything together inside a cell is metabolism. Where is energy stored in the ATP chemical? So ATP has 3 phosphate groups, and the bonds between the phosphates is where majority of the energy is stored. When ATP breaks off one phosphate, it releases energy. Number 5, what happens to a cell if it's placed inside a hypertonic, hypotonic, or isotonic solution? Well, this depends on the type of the cell. If it's a plant cell in a hypertonic solution, so this is when the environment has more salt or more solute. So think about salt, right? So more salt in the environment, therefore relatively less water in the environment, therefore water will go out of the cell membrane to enter the environment. So therefore the cell membrane will shrink, causing the cell membrane separating from the cell wall. So if it's a plant, then the plant will wither. In a hypotonic solution, water will enter the cell because hypotonic solution has less salt, so therefore relatively more water. The water will go into the cell, causing the cell membrane pressing against the cell wall. But because plants have cell walls, so therefore plant cells will not burst. Plant just will become really turgid. In an isotonic solution, water molecules can move back and forth freely, equally, so therefore you won't notice much effect. In an animal cell, this is different. The main reason is because animal cells do not have a cell wall. So therefore, in a hypotonic solution, too much water may enter the animal cell, causing the cell membrane to expand too much and possibly even burst, therefore killing the animal cell. In a hypertonic solution, of course, the animal cell will shrink. In an isotonic solution, there's no change to the size of the cell. Number 6, how are the 3 mechanisms of endocytosis different from one another? So there is pinocytosis, that's when liquid or things that's dissolved in the liquid, in the fluid that enters the cell. Phagocytosis is when solids, particles, etc., bacteria, viruses, when they are brought into the inside of the cell. And then receptor-mediated endocytosis refers to those specific substances that can be recognized by the receptor cells on the surface of the host. So basically, it's specialized substance brought into the cells. Number 7, how do endergonic reactions and exergonic reactions differ? Is a catabolic reaction exergonic? Endergonic reactions means that the chemical reaction, in order for that to take place, it needs some energy input. It absorbs energy. Exergonic reactions releases the energy that can be used for other activities in the cell. Catabolism means the breakdown of large chemicals, break down into something simpler, so therefore it's going to release energy. So therefore, yes, it is an exergonic reaction. Number 8, molecules across the cell membrane through different mechanisms. How do water, carbon dioxide, and glucose cross the membrane? Where water is osmosis. Osmosis describes the movement for the water only. Carbon dioxide will easily diffuse to the inside or the outside of the cell based on its concentration, because it's small enough and also because it's nonpolar. It has similar properties as the phospholipid bilayer. So it's simple diffusion for carbon dioxide. Glucose will also follow the concentration gradient, but its diffusion needs the help of the proteins that's embedded in the membrane. So glucose will cross the membrane through facilitated diffusion. Are these active or passive transports? Because all of them will move with the concentration gradient. So therefore, it is a spontaneous process it does not require energy. Those are considered passive transports. Chapter 6. First question. Where do light-dependent and light-independent reactions happen in the chloroplast? Light-dependent reactions splits water and produces oxygen. This happens in the thylakoid membrane. And the light-independent reactions happens in the stroma. These involve the steps that mix glucose. Second question, what colored light has more energy? Lights with shorter wavelengths have more energy. That means the violets have the most energy. The color that you see in an object is due to the light absorbed or the light reflected. So remember that if the light has been absorbed, then you won't see that color. So therefore, it's the light reflected. Number 3, how do photosystem 1 and 2 replace their electrons? So first remember that photosystem 2 happened first and then it's photosystem 1. In photosystem 2, the electrons lost is replaced by the electrons from water. And in photosystem 1, the 2 lost electrons are replaced by the electrons passed from photosystem 2. Number 4, what molecules are produced by photosystem 1 and 2? In photosystem 2, oxygen will be produced, ATP will be produced. In photosystem 1, most of the time, the only product is NADPH. Number 5, overall, what are the products of photosynthesis? The products will be glucose and oxygen. Think about photosynthesis being a process that plants make sugar and make oxygen. Chapter 7. First question, what are the products of aerobic respiration? Aerobic respiration is almost the opposite of photosynthesis. So the products are carbon dioxide. When we go through this aerobic respiration process, we exhale carbon dioxide. So that's one product. The other product is water, which we call the metabolic water, right? So CO2, water, but also a lot of energy. From one glucose, we can make as many as 36 molecules of ATP. Second question, what are the 3 stages of cellular respiration? Stage 1, glycolysis. Stage 2, Krebs cycle, which also has a transitional step that's called acetyl-CoA formation. And then stage number 3 is the electron transfer chain. Which step is the oldest evolutionarily? That would be the first step, the glycolysis. Where does each step take place in a eukaryote? Glycolysis happens in the cytoplasm. Krebs cycle happens in the mitochondrial matrix. And electron transfer chain happens in the inner mitochondrial membrane. Number 3, where do the spent electrons from the ETC, the electron transfer chain, in the aerobic respiration go? So those electrons, after passing through those many proteins in the ETC, will eventually reach the electron acceptor, oxygen. So the answer here is oxygen. Number 4, can human ferment? Yes, we can. We can ferment. We can go through this lactate fermentation. And if so, where does it take place? It normally happens in these white muscle fibers. So these fibers do not have a lot of mitochondria. Therefore, they cannot produce energy using aerobic respiration efficiently. At times when we need some quick spur of energy, those cells will go through lactate fermentation and produce some quick energy. Chapter 8. First question, which nucleotide is not found in RNA? So basically we learned 5 different types of nucleotides. There's adenosine, there's guanine, cytosine, thymine, and uracil. RNA does not have thymine. No T. Number 2, what is the contribution of Griffith, Watson-Crick, Everett McCarthy to the discovery of DNA. Griffith is the scientist who first discovered that a non-pathogenic bacteria can become pathogenic, so basically can take DNA and become transformed. Watson Crick discovered the structure of DNA. Of course, there's also Franklin. Everett and McCarthy, they're the first ones to confirm the function of DNA. They confirmed that DNA is the hereditary material instead of protein. Number 3, you should be able to replicate DNA and transcribe DNA to RNA. When you replicate DNA, make sure that you not only match up the letters, right? If you see T, you put A there. If you see G, you put C there. But also note the directions. If the DNA runs from 3' to 5', and then the replicate, this complementary sequence should run from 5' to 3'. When you transcribe DNA to RNA, you follow the same rules, except that when you see A, you're going to put a U there instead of T. Chapter 9, what is an intron? What is an exon? An intron is a sequence that will not be used to make genes. These are the interference sequences, the sequences that's inserted into the genes. What is an exon? The exon is the expressed portion of the gene. So the letters in the exon will be used to make proteins. How many nucleotide letters correlate to one amino acid? The answer is 3. Chapter 10, question number 1. What type of tumors are cancers? There are 2 different types of tumors, malignant and benign. So the benign ones, they do not cause cancers. They're harmless. The malignant tumors, they will metastasize and they will cause cancers. Number 2, what is the function of telomerase? The function of telomerase is to maintain or extend the sequence of the telomeres in a chromosome. So they're going to add the letters back to the end of the chromosomes to maintain the length of the telomeres, which has something to do with the life of a cell, which is why cancer cells are immortal, because their telomeres never shorten. Number 3, what is the Hox gene and what is the Zist gene? Hox gene is a master gene. This gene controls the body planning. Basically, it tells the cell when to produce a body structure, how many parts that structure should be produced, etc. The Zist gene, XIST, is a gene that causes X inactivation. So basically, one of the 2 X chromosomes in female cells will have the Zist gene transcribed into an RNA, and this RNA will then cause that X chromosome to condense into a bar body to become deactivated. Chapter 11. What happens during each of the 4 mitotic stages? So first remember that the name of the 4 stages are prophase, metaphase, anaphase, and telophase. In the prophase, first the DNA will condense, the sister chromatids will become visible, spindle fibers will form, spindle fibers will connect to the sister chromatids on the 2 sides, and also the nuclear membrane will disappear. In the metaphase, spindle fibers will align all of the sister chromatids in one straight line at the middle of the cell. At the anaphase, the 2 spindle fibers attaching to one pair of sister chromatids will pull the sister chromatids apart towards the 2 poles of the cell. In the telophase, the sister chromatids have already been separated. They will reach the end of the cell, new nuclear envelope will form, and then the sister chromatids will decondense. And then the cell will also get ready for cytokinesis. Number 2, what's the longest period of time in a cell cycle? So a cell cycle, remember that the longest time of the cell cycle is the interphase, where you do not see much activity underneath the microscope, but in fact the cell is most active. And that interface also can be divided into G1, S, and G2, 3 different steps. Chapter 12. 1. Do body cells have sex chromosomes? The answer is yes. All the somatic cells in the human body have the exact same genetic makeup. All of them have 46 chromosomes, 23 from father, 23 from mother. Number 2, how many autosomes and sex chromosomes does a human gamete have? A gamete has half the number of the chromosomes that's found in a regular somatic cell. So altogether it's 23, but that 23 can be put into 2 categories, 22 being autosomes and 1 being the sex chromosome. Number 3, when do homologous chromosomes cross over during meiosis? That would be prophase 1. Number 4, what is the SRY gene? SRY gene is the sex-determining region gene on the Y chromosome. This is the gene that tells the embryo, remember that in human, males and females start with the same genitalia that looks like female, but at a certain stage of the development, the SRY gene will cause the male fetus to produce testosterone. This hormone will then cause the male embryo to dismantle the female structure and start to grow testes and other male genitalia. Chapter 13, question number 1. Who is the father of genetics? That would be Gregor Mendel. What organism did he study? He studied garden peas. Number 2, what are the 2 laws of Mendelian genetics called? The first law is called the law of segregation, and the second law is called the law of independent assortment. Number 3, what is a phenotype and what is a genotype? What's their relationship? The phenotype is the outward expression of a trait. You can see that directly with your eyes, or you can sense it, you can smell it, for example. It's the observable trait. What is genotype? Genotype is the genetic makeup, is the allele combination for that trait. Their relationship is that the genotype will determine phenotype. Number 4, what does heterozygous mean? Heterozygous means that the 2 alleles that's found in the individual, they are not the same. What is a carrier? A carrier means a heterozygous individual for a recessive trait. What type of diseases have carriers? Only recessive disorders have carriers. That means autosomal recessive and X-linked recessive. Number 5, you should be able to solve Punnett square problems to predict offspring phenotypes. If you go all the way to the end of the slide, there is a Punnett square exercise. Black fur in guinea pigs is dominant over white fur. Find the probability of an offspring with the same phenotype as its parents in a cross between a homozygous black and a heterozygous black guinea pig. So it should be simple. So you're going to write 2 capital B's on the top for the homozygous black parent, and then you're going to write a big B and a small b on the left side for the other black furred guinea pig parent. And then put the combinations in the Punnett square, and you will see that all of these guinea pigs in the second generation will have a capital B. This capital B because it's dominant, so therefore all the offspring will be black. The question asks you the probability of offspring with the same phenotype, right? The phenotype is basically being black or being white. So therefore 100% of them will have the same phenotype. So now go back to chapter 13, question number 6. What's the difference between polygenic inheritance and pleiotropy? Polygenic inheritance tells us that one trait is determined by many genes. Poly means many, genic from genes, right? Many genes determining one trait. Pleiotropy is the opposite. This is one gene, and this gene is important. For example, the gene that's responsible for producing the connective tissues. Mutation of that one gene can cause many manifestations of diseases, such as Marfan syndrome. One gene determines many traits is pleiotropy. Number 7, people with what type of blood are universal blood donors? That would be type O, because type O do not have either antigen A or antigen B. Universal acceptors would be type AB, because they already have those antigens in their body, so therefore they are not afraid to take in more A's or more B's. Question number 8, which type of blood are definitely homozygous genotype? That would be type O. Type O must be 2 lowercase I, right? The allele combination must be lowercase I, therefore homozygous. And which one for sure is heterozygous? That would be type AB. That would be capital I with a superscript A and a capital I with a superscript B. Number 9, you should be able to predict the blood type of a child if a parent's blood types are given. So again, this is a Punnett square problem. Refer to that slide in chapter 13. Chapter 14, question number 1. What chromosome aberrations, so basically changes, are possible during crossing over? Think about duplication, deletion, inversion, translocation. What is translocation? Translocation refers to the portion of one chromosome is swapped with the DNA that's found on an entirely different chromosome. So remember that crossing over should happen between homologous chromosomes, but in translocation, they happened on 2 entirely different non-homologous chromosomes. Question 2, what is a non-disjunction error or aneuploidy? Non-disjunction error means that one chromosome has excess amount or has fewer numbers than how it should be during meiosis. So basically either meiosis 1 or meiosis 2 had some mistakes in separating the chromosomes. What type of diseases can it cause? It can cause either trisomy or monosomies. Some examples include the Down syndrome, which is trisomy 21, Turner syndrome, which is XO, missing one X chromosome, Jacob syndrome, XYY, Klinefelter syndrome, XXY. Number 3, what are the major characteristics of autosomal dominant, autosomal recessive, and X-linked recessive diseases? First, if it is autosomal dominant, you should see equal occurrence between males and females. You should also see that the disease does not skip generations when you go backwards. If one person is affected, there must be at least one affected parent. Autosomal recessive disorders, if you see that the disease skips generation, it must mean that the disease is recessive. Because it's autosomal, so therefore you're going to see the disease affecting males and females equally. X-linked recessive disease will affect mostly just the males. Females tend to be carriers. And also remember that for X-linked recessive disease, males will never be carriers. And if a female is affected, this female must have an affected father. When I gave you a pedigree chart, you should be able to determine the type of inheritance using the process of elimination. So now, again, let's go to the exercise on the second to last page. There are 3 exercises. The first one, if you look at the first one, you can see that males and females are affected equally, so therefore this must be an autosomal pattern because it doesn't seem like the disease skipped generations, so therefore it could be autosomal dominant, but it can also be autosomal recessive. But let's see that it's autosomal recessive, and then that means that, father here has 2 lowercase letters. Yeah, mother may have 1 lowercase, 1 uppercase, or 2 uppercase. It would be possible. Yeah, so either dominant or recessive, it can make sense. So I don't think there is a definitive answer for the first one. So it can be autosomal dominant, but can also be autosomal recessive. For example, if mother is heterozygous, and then some children will be affected, others will show the dominant phenotype. The second pedigree chart clearly is skip generation, right? It's because this child does not have an affected parent. So therefore, this must be a recessive disorder. And then we also see that only males are affected. So this must be an X-linked recessive disorder. The third one, males and females both affected, and the disease skip generations. So therefore, this is the autosomal recessive disorder for sure. Only the first one, you can't see for sure if it's autosomal dominant. So now go back to Chapter 14, question number 5. Can a woman get an X-linked recessive disease? If so, how? So again, if a woman gets an X-linked recessive disease, she must have 2 bad X chromosomes. Again, she will get 1 from father, 1 from mother. If a mother has 1, mother could be healthy, mother could be affected. But if father has a bad X chromosome, father must be affected. So basically, she must have 2 bad ones, and then the father must be affected. Chapter 16, question number 1. Who discovered the evolutionary theory by natural selection? There are 2 scientists who discovered it. Remember Charles Darwin, also remember Alfred Russel Wallace. Question number 2. What is a vestigial structure? This is structure that's fully functioning in some animals, but not functioning in the species that you're studying. Question number 3. What eon, era, epoch, and period are we currently living in? So we're in the Phanerozoic eon, the Cenozoic era, the Quaternary period, and the recent epoch. How does radiometric dating work? So radiometric dating is based on our knowledge of the half-lives of these radioisotopes. Half-life is the time that it takes 50% of the radioactive isotope to change into a different species. If you know the half-life, you can calculate roughly the age of a fossil. For example, if you look at this question, an elephant fossil was found. Chemical analysis revealed that it has 12.5%. 12.5% basically is 1/8. 12.5% of the C14 found in a living elephant. How long ago did the elephant die? So basically, when scientists unearthed the elephant, only 1/8 of the C14 is left. 1/8 is 1/2 to the 3rd power, That means that 3 half-lives have passed after this elephant died. And because we know that one half-life for carbon-14 is 5,730 years, so therefore this elephant died 5,730 years times 3. That would be 17,190 years ago. We have some more examples in the PowerPoint slides, so go back to the slides and work on those exercises. Chapter 17, question number 1, how is allele frequency calculated? So again, the most important thing is to remember that when you have n individuals, you will have 2n number of total alleles. So the allele frequency is the type of allele that you can count in a population divided by twice the number of the population. For example, the question over here, 10 individuals having 4 dominant alleles, the frequency of the dominant allele will be 4 dominant alleles divided by 10 multiplied by 2, 4 divided by 20 basically, right? Because 10 individuals, 20 alleles. 4 divided by 20, that would be 20%. So the allele frequency for the dominant allele is 20% here. Question number 2. How do you determine if the daughter generation has evolved from the parent generation by using the Hardy-Weinberg equation? Basically, the Hardy-Weinberg equilibrium suggests that if a population shows no evolution, then the allele frequency does not change. So therefore, you will calculate the allele frequency in these different populations, and if you do not see a change, then there's no evolution. So again, if you do that, if you track the allele capital A, the allele frequency for capital A in the parent generation will be 1, 2, 3 divided by 8, 3 over 8. And then if you look at these 4 different choices, population A also has 3 capital A's over 8. So allele frequency didn't change, no evolution. For second population, you see 1, 2, so therefore no evolution. The third one, 1, 2, 3, 4 over 8, so there is evolution. And the last one is 1, 2, 3, 4 as well, 4 over 8. So again, it's different from the parent allele frequencies. Therefore, B, C, and D all have evolved. Number 3, how do you use that equation to find out the percentage of disease carriers if you know the incidence of a recessive disease in the population? So again, the incidence of a recessive disease basically is the percentage of the people that are affected. Basically, that's Q squared. So if you know Q squared, then you can figure out what Q is. And once you figure out what Q is, you can figure out what P is, because P plus Q equals 1. And once you figure out what P is, you multiply 2 by P by Q to figure out the percentage of disease carrier. So 2PQ is the percentage of disease carrier. Refer to that exercise in the chapter 17 slides. Number 4, what are the 3 modes of natural selection? That would be directional, disruptive, and stabilizing. Number 5, what's sexual dimorphism? Males and females showing different morphotypes. What's a balanced polymorphism? That means that many different forms for one trait. And even though some traits are not favorable, but they exist because there are some other advantages, so therefore they're balanced. Heterozygous advantage is one example for balanced polymorphism. Heterozygous advantage basically tells us that some disease carriers can survive better in certain environments. So therefore, those disease carriers are favored by the environment, by natural selection. Number 6, how do the three geological speciation patterns differ? Allopatric, that means the two species are found on the two sides of a geological barrier. Parapatric, on the same side. And then the last one is sympatric, that these species are mixed very well in the same environment. Example for allopatric, think about the snapping shrimps. For the parapatric, think about the giant velvet worms. For sympatric, think about the fish that lives in that lake in Cameroon. Number 7, how does polyploidy help plants to evolve new species in an instant? So think about the cotton that we grow today, right? The cotton came from the Old World cotton plant and the South American cotton plant. Both of them have 26 chromosomes, but the modern cotton has twice as many. So basically, a meiosis error caused these two plants to combine. Because the polyploidy plant can mate with neither of its parents, so therefore it must be a new species. Number 8, what is the difference between the bottleneck effect and the founder effect? The bottleneck effect is when the population significantly decreases, that very few alleles are left. The founder effect does not have this sharp decrease of the population, but instead, a few individuals in the main population leaves the main population and starts to reestablish in a new place. Number 9, what does prezygotic reproductive barriers mean? And what are the possible types of pre- and post-zygotic reproductive barriers? Prezygotic reproductive barriers basically means that these are the barriers that will prevent the formation of a zygote. There are 5 different types of prezygotic reproductive barriers. That would be the ecological, temporal, behavioral, mechanical, and gametic. Make sure that you go back to that slide where you see those examples. Post-zygotic reproductive barriers have 3 different types. The first one is hybrid inviability. Second one is hybrid infertility, and the last one is hybrid breakdown. So again, refer to that slide where you can see the pictures. Chapter 18, question number 1. You should be able to apply the rule of simplicity to identify the most probable model, probable tree of evolution. So again, you basically put those characters onto these possible trees and count how many evolutionary events each tree have. The tree that has the least number of evolutionary events will be the most probable mode of evolution. Question number 2, what is a clade? A clade is a group with some shared inherited characteristics. So this could be a lineage of species. It can also be the 2 sister groups plus the common ancestor. So therefore, you can consider a cladogram being a clade inside clade. Number 3, how do you differentiate homologous structures from analogous structures? Homologous structures, they are made of the same materials, but they may have different purposes. Analogous structures, they are made of different materials, but have the same function. So these two are opposite to each other. Number 4, you should be able to read and interpret cladograms. And number 5, what are sister groups? Sister groups are these 2 branches joined together at one single point. And sister groups are normally closely related. So if we go to this cladogram on the last page of the study guide, you will see the clam and the earthworm being sister groups, starfish and human being sister groups, and you will see the starfish is a clade, human is a clade, starfish, human, and their common ancestor belongs to another clade. You will see that the starfish is more closely related to human than to the clam, because the common ancestor of these two is over here, closer to the tips of the branch than the common ancestor between the starfish and the clam, which is over here. Chapter 19, question number 1. What molecule is a requirement for life as we know it? The answer is water. Water is essential for anything that we know of that lives on the earth. Second question, what are some of the hypotheses regarding the origin of life on Earth? The first hypothesis says that life likely started from the Earth with the lightning catalyzing these chemical reactions, synthesizing organic chemicals from inorganic chemicals. Remember that Urey-Miller experiment? The second hypothesis states that life started from the hydrothermal vents at the bottom of the ocean. The third hypothesis basically says that these organic monomers that's required to build life come from outer planets. Question number 3. What biological chemicals were produced in Stanley Miller's prebiotic assimilation experiment? The answer is amino acid. Question number 4. Why is RNA hypothesized to be the first form of genetic material? The answer is found on slide number 20. So basically, RNA has 4 specific properties that makes it most probable form of first genetic material on the Earth. Number 1, RNA can store genetic information. Number 2, it can make proteins. Number 3, some RNAs can act as catalysts or enzymes. Number 4 is that RNA can duplicate itself. So therefore, very likely it is the first independent precursor to life on the Earth. Question number 5, what is the endosymbiont theory and what are the evidences? You can easily find the answer on slide 30. This theory basically states that mitochondria and chloroplasts, they evolved from bacteria and they used to be free-living. Over the time, their host cells become dependent on each other. Their host cells provide a safe and stable environment for them, and they provide either energy or photosynthesis for the host cells, basically getting food and energy for the host cells. The evidences include, number 1, mitochondria and chloroplasts, they have similar sizes and shapes, as well as membrane compositions to bacterial cells. Number 2, they both have double membranes. The inner membrane is their initial membrane, and the outer membrane is the membrane from the host cells during that endocytosis event. Number 3 is that both of them can divide independently. Number 4, chloroplasts have pigments that are similar as the pigments in the cyanobacteria. Number 5 is that they both contain their own and single circular DNA. They also have their own RNA and ribosomes that are similar to bacterial ribosomes. Number 5 is that if you compare their DNA sequences, they are very much similar to the DNA sequences of some known bacterial species. They do not have introns. Again, a feature that's found in mostly just prokaryotes. Question number 6. What is the order of evolution of life on the Earth? So basically, you can refer to slide 33 of chapter 19. There are 9 steps. Step number 1, building blocks formed. Think about these proteins and lipids, polysaccharides, and RNAs form. Number 2 is cells formed. Think about the first protocell assembling all those biomolecules together into one entity. Number 3 is that domains start to diverge. Besides bacteria, now we also started to have archaea. Number 4, photosynthesis and aerobic respiration evolved. At the beginning, photosynthesis only happened in bacteria. Aerobic respiration also happened in some bacteria only. And number 5 is endomembrane system and nucleus evolved, which started to produce eukaryotic cells. Number 6 is the origin of mitochondria. Some aerobic bacteria was swallowed by some archaeal cells. Number 7 is the origin of chloroplast. Some of these eukaryotes with mitochondria then engulfed some cyanobacteria, which developed into chloroplasts. Number 8 is the origin of fungi, animals, and plants. And finally, the modern lives evolved. So those are the 9 steps. You should roughly know this order. Good luck to you for your final exam. If you have any questions, please feel free to email me.